All right, good morning, everybody. Any questions, concerns? Okay, we left off about halfway through this chapter, I think, pretty close. So with that, I don't think I described anything on here. We talked a little bit about some gene therapy and possibilities with cures and ultimately some setbacks with that as well. There are other ideas when we think about therapy as a form of treatment, and that's definitely more, at least at this point, more of a possibility. So it really, when it comes to some of this, kind of depends on the circumstances as far as how we might be able to deal with some of this in the first place. And a lot of the time, we think about utilizing a virus that isn't infecting us with a virus, but infecting us with a good copy of a human gene. So it's basically just being used as like a vector, as this vehicle to transport this genetic information into a defective cell. The virus has different, again, depends on the type of virus, but the viruses have different potentials as far as the cell that they might be infecting. So if you get a virus that can make that specific cell its target, you're able to infect it, again, with not viral stuff, but the good stuff, the good copy, if you will. So that's what we've got up here is this adenovirus, finding the specific cell, the target, docking with it, with its specific spike proteins. Lots of viruses have kind of this fingerprint that allows them the specificity to target a respective cell. So that lends to a greater likelihood of the target DNA getting into the right stuff and not winding up someplace where it shouldn't be. There are still potential issues as we think about maybe the right amounts, the concentrations of these things getting into the respective cells, and that sets the stage for some potential issues, right? So there's all sorts of things when we think about manipulating stuff, importing a different type of a gene into something like these mice that have a fluorescence gene in them. A black light, a UV light, causes them to glow. You can see the tail, the eyes, the nose, all lighting up based off of this. So the middle guy is not altered or manipulated. It's the control, if you will. So it looks like a normal little lab mouse should look, all right? Just a little white mouse. You can see its tail and its eyes. They're not all lit up, all right? So these have something different going on, this fluorescence gene, and we would call them transgenic, right? Because they got something going on. So with that, this has nothing to do with the mice, but it is kind of fits right in this realm as we think about it. I saw an article. I did not read through all of the details. I have heard of all sorts of potentials, usually with pigs. Pigs and people genetically have a lot of similarity. And there are things like maybe a heart valve that gets harvested out of a pig and you can transplant that into a person and you have very little issues with reactions. There are other thoughts where you can create kind of a transgenic situation. So you put some human genes into this pig and now it's expressing these things. And so it's more similar to human than just the pig by itself, transgenic, right? So the thing that I saw was a pig that grew a human kidney And that human kidney was transplanted into a person with basically no issues of rejection, at least so I gathered from the brief little bit that I glossed over it. And the kicker is, it's not rejecting it because it would be like my genes for this put into that pig. So when it makes the kidney, it's my kidney. It's got my own fingerprint in it, my molecular fingerprint, not my fingers, right? But it says, this is mine. There is no rejection like that. That is awesome stuff. And the potential for something like that, because organ transplants set the stage for all sorts of issues with immunosuppressive drugs to limit the amount of the body's potential to try to reject those organs. If your body gets an organ that says this is my own kidney or my own liver, even though it was grown in a pig, rejection isn't an issue. It's your own stuff. And I think that is pretty awesome, right? Not the same, but kind of same sort of an idea. And that pig would be transgenic, right? It's got altered stuff within it. So there are all sorts, when we think about biotechnology, when we think about engineering stuff, we could look at agriculture and not think about doing stuff in a lab. That's definitely a possibility. But if we step back to the dawn of agriculture, we have been manipulating stuff since we started trying to grow crops or to raise livestock in the first place, right? You keep these seeds that were, more robust, that were able to be, maybe a little more drought tolerant or something like that. But really, we think more like greater yield, more kernels of corn. That's something that might be an advantage. More food stuff, right? More bang for your buck. You get a cow that is bigger and produces more milk. Maybe that's the type of cow that we would like to have other cows that are similar, or a bigger beef cow that we can get more meat out of. Something like that we can select for, and over the course of generations, enhance some of those specific characteristics without ever stepping into a laboratory situation, right? Just by, cross-pollinating or breeding these animals together. We could look at dogs as a great example with all of the diversity we see in dogs, all ultimately descended from wolves that look very similar. But we start selecting specific traits that we find, whatever, appeal to it, the size, the shape, whatever, right? And we select for those types of things, right? So without laboratory situations, biotechnology, the manipulation of genomes has been occurring for a long time, right? We step it up and start thinking about other possibilities, knowing some of these details, some of the genetics behind this. We can use that to our advantage, where we can see crops that are manipulated that lend to stuff like maybe, pesticide or herbicide resistance. Maybe that's good, maybe it's not, all right? That kind of just depends on the circumstances. There are also issues with some of these things, because if we're thinking about incorporating something into a plant like corn, as we've got up here as an example, there is a type of pesticide that uses some bacteria components, right? It's a type of crystal, almost like a shard of glass, that these bacteria can produce just in part of their normal life cycle, right? But these substances, if we take that, extract it, and just use that fragment of the bacterium, it's a pretty stinking good pesticide, right? And it's been incorporated into pesticides for decades, right? This little crystalline structure from a bacteria, right? So it's been used quite a bit, right? And it's been put into stuff that's sprayed onto crops. But the bacteria is a living organism. It's got genes that say, build me this crystalline structure. We can take that gene from the bacterium and we can incorporate that into a corn plant, creating transgenic corn. And this corn, as it grows, it also produces that specific protein, that crystalline structure. Now the corn plant, we don't spray it with pesticides. You don't have to, because the corn plant's producing the substance that kills the bugs within its leaves. So it's not something that the bugs are going to avoid it. They're going to come and try to eat it. And as they eat it, they ingest that and then they die. All right. So seems like maybe a pretty good idea, right? You could incorporate that in there and we can now limit the amounts of stuff, the amounts of crap that gets sprayed on all of these crops. Maybe, maybe not, right? After a handful of years of this growing in the fields, we see, just like, you know, the overuse and misuse of antibiotics lending to big drug resistance with some of these microbes, the mass quantity of these crops that are grown with this specific thing in it sets the stage for some insects to have some sorts of tolerance, some sorts of resistance. And over the course of time, that compounds. We add that all together, and all of a sudden, you've got a huge population of insects that no longer care about this type of gene, this type of crystalline structure. It does nothing to them. So they're able to feed on these corn plants and decimate the crops, right? Corn farmers wonder, as they're looking at these fields, this wasn't supposed to happen. Yet all of my corn plants have been knocked over as these borer beetles have chewed through the stalks of all of the corn plants. That's an issue, right? So sometimes advantage. Sometimes these things that we think of as being an advantage may blow up in your face, and that's definitely an issue as well, right? We do see other things that potentially lend to greater output, higher yield, things like that, that we might think of as advantages. But also there are things that we think of as a solution that may only kind of be a Band-Aid and not really the solutions that we're hoping for. Sometimes things like, you know, herbicide resistance gene just makes it so that these crops are able to be sprayed with more chemicals because those don't harm the plant. And that begs the question, what does that do to the food chain, to the supply? What does it do to you as you're consuming all of these things, right? And we're definitely recognizing all sorts of potentials with different types of molecules that potentially get into the food supply and persist. These forever chemicals that beg the question, do you know what's in your food? Maybe you don't want to know. It could be a little disturbing. So this, as we're looking at this, this is just a map of one of our chromosomes, so X chromosome, and it's showing all sorts of different genes. We've got all of these tagged on here, and there are some potentials as we move through here and look at a couple of X-linked or genetic diseases that are passed on these X chromosomes, right? Fanconi anemia, Wiskott-Aldrich is, I mentioned the SCID, the severe combined immunodeficiency. Wiskott-Aldrich is kind of like that, where you've got some issues with immunity, with some of your immune cells, and are more susceptible to infection. These individuals, if I recall properly, don't make it out of puberty. They die before due to all of these issues. All of these things really, as we're looking at this, beg the question, can we target this? Could we go flip back to that picture of the virus that's targeting a cell? Can we bring in specific genes that might help to alleviate this problem in the first place? And Wiskott-Aldrich is definitely one, like the SCID, that we have used these genetic techniques to put the proper copies in, and we have cured a few individuals. It also is one, just like SCID, that we start thinking more therapy stuff, not curing these people, but pulling out specific cells, targeting that, correcting the cell, but not the person in general, right? And that requires basically prolonged therapy, treatment every handful of months to deal with it. But that's a better situation than the outcome you would receive with nothing. So when we think about studying things, there are all sorts of organisms that we think about as what we call model organisms. These are organisms that have a tremendous amount of research done on them, sequencing genomes, all of these things. Typically, we've got all sorts of this information at our fingertips. And the ability to work with and manipulate some of these things sheds light on all sorts of other organisms, potentially ourselves, right? As we think about some of these things, right? So here we've got a bunch of different organisms. Our little mouse, Musculus, fruit flies right here, Drosophila. This is a type of nematode, Cinerorhabditis elegans, C. elegans. Saccharomyces is a type of yeast, Saccharomyces cerevisiae yeast that we think about for beer, but also bread is a type of cerevisiae yeast. And this pretty little flower, Arabidopsis, is another one when we think about plants, a model organism. I recall, and all of these, scientific names. When it comes to plants, I want a common name. I have difficulty knowing all of these specific things. So there are seminars that happen with graduate students talking about their research. They do proposals and then they do defenses. So they set the stage. This is what I'm hoping to study. And then they defend it after a handful of years and say, oh, these are my findings. This is what's up. So I've studied microbiology. I studied a lot of really fungal organism and some immunology behind that. So not really thinking botany, all right? But I sat through numerous presentations, numerous seminars where students are talking about studying Arabidopsis and all of this stuff, right? All of these details about Arabidopsis and how cool it was, how you're able to manipulate it. You can do this, you can do that. You can genetically modify it and see what happens when you've done that, right? And I don't want to be the guy in the audience that's like, what in the hell is Arabidopsis, right? It sounds cool, but I don't know what it is, right? So after a handful of these seminars, I decided I'm just going to look it up. I'm going to see what is so stinking cool about Arabidopsis. It looks kind of interesting, all right? And as I do a little digging into it, it's a wild mustard. It's like a type of mustard that you'd see growing as a weed on the side of the road, all right? Now I'm not so impressed, right? I'm like, stinking wild mustard? That's what you guys have been talking about all of this time and saying how awesome it is, and it's a weed? Okay, right? It's still interesting, right? And it is a model organism, right? Drosophila, these little fruit flies, we've used for all sorts of details and research. And I think it's pretty interesting, right? I've got a couple of examples with the Drosophila that show, I guess, targeted evolution. It's change, but it's not change of all fruit flies. It's this one laboratory population of fruit fly, right? You're not changing them all. just this group you're working with, right? It's a controlled situation. And here we can see variation in the first place with fruit flies with red eyes and some white eyes. There's variation and variation in the wings and whatever, right? This one study looked at the longevity of a fruit fly, right? And basically, the little fruit fly, once it goes from the, maggot stage to the adult fly, really the idea for this fly is to find food, to eat, to breed, to lay eggs, and to die. They don't live for a super long time, right? And they need to eat so that they've got ample energy for reproduction in the first place. So they pump out a bunch of eggs, right? So they wanted to see what would happen with these fruit flies, and could we change this population of fruit fly? Could we make some that are able to tolerate long periods of starvation? And in order to do that, you got to kill a bunch of fruit flies, right? So they had some containers, like big aquariums, that are all sealed up, that just the flies are stuck in there, they can't get out, and other flies can't come in, right? And they get these adults and they take away the food supply and they watch them die. And you kill off basically about 80% of these flies. It only takes about a day, right, for these flies to go without any food, and then they can't survive, right? So you start with, thousands, 5,000 or so fruit flies, and you kill off 80% of them. So now you got a handful left, right? You take the food, you put it back in, you feed them, you breed them, they lay their eggs, and this population grows up, right? And you do the exact same thing. As soon as we hit the adult stage, pull the food away and watch them drop off, right? Fruit flies reproduce very rapidly, right? And I said adult state, we can live about a day, right? They do this for about 60 generations, right? In human time, that's a lot of generations, right? In fruit fly time, well, we can do this in a handful of months, right? That's not that much time to get 60 generations in a fruit fly, right? It's a boatload of time to think about people, all right? So 60 generations later, after we have done this process, we pushed it from being able to survive for a day to like a week and a half, if I remember the numbers right, right? That is crazy to think about the potential to do that, right? There have been other studies with fruit flies, again, thinking about feeding them ethanol, alcohol, right? Then I'm like, well, that's a bar fly, not a fruit fly, right? That's a little bit different. It's the same stuff, right? So these fruit flies, technically, alcohol is not something they really want to consume, all right? It's not something that they're thinking, oh, that's what I want to tap into, all right? And there are some fruit flies, just like this longevity deal, there are some fruit flies that could tolerate a little bit of alcohol. So we select for those. The other ones can't, they die. That type of food's not good for them, right? But the small select few that are able to tolerate it, they breed, they ultimately give rise to another generation that's a little more tolerant. And over the course of multiple generations, you push the threshold, the tolerance from low concentration to pretty high concentration of alcohol, and these flies can feed on it. Crazy, right? As you focus in on the specific metabolic genes that allow them to deal with that type of stuff, right? So all sorts of research, right? And it really kind of just depends on the circumstances, right? We don't have any bacteria up here, but E. coli is also a model organism, a type of bacterium that is used very frequently in genetic engineering and manipulating things. And it's also, like this, where we know all sorts of details, sequencing genome and all of that to shed light on, well, on whatever. So pretty interesting to think about, all right? So Metagenomics is looking at like a specific area, specific habitat, type of environment, and all of the things within that habitat, in that environment. So multiple species. We isolate DNA from multiple things within that sampling area, and we see what sorts of stuff is there, what sorts of genes are present, what sorts of capabilities do these things have. And it might be, we can reduce it down a little bit and think about, this little plot of soil and what sorts of microbes are in this soil. Bacteria and fungi are some of the key ones that we think about, but there's other stuff as well. There's protists, single-celled eukaryotic organisms in there. And in this sampling of soil, there's all sorts of potentials for them to be interacting, to be competing, or to be working together. And metagenomics sheds some light on some of the specific details that all of these organisms hold, again, within this specific area, this little sandbox full of soil, right? So here we've got different genes, DNA from different species, and we take this and we'll use those molecular scissors that we talked about before, those restriction endonucleases, same scissor, different species, right? And we take those scissors and apply them to the genome, to the DNA, and chop it up to see what sorts of fragments do we get, right? Different looking patterns, different snippets of DNA, variation, right? Different capabilities, different potentials to survive or to try to inhibit somebody from surviving within this type of habitat. And with that, you can create these libraries that show some details and some genes that are shared. Also the opposite, things that are not shared, novel and unique things that ultimately set the stage for diversity within that, right? We think about our own body, before I shift over to all this algae, we think about our own body and like the human genome, looking at all of our DNA. But then we think about the microbiome and all of the organisms that live on and in us and all of their DNA, all of their information. And the metagenomics can look at that and look at all the plethora of organisms that we've got calling us home and what sorts of capabilities that holds, right? We're learning all sorts of things about gut bacteria, organisms that thrive within our stomach, our intestines, within the GI tract that set the stage for all sorts of shifts, changes, and cues to our behavior that are all directed by the crazy microbes that live within us. And some potentials for disease we are recognizing might be the profile of microbes that you've got within your gut. So, they say you are what you eat. And I think maybe that's pretty stinking true as we start thinking about what types of things we are putting into our body. We should be thinking about who we are truly feeding. It's more than just ourselves, right? There's others, and that imparts all sorts of potentials within us, right? Pretty crazy, right? And there's some examples, people that have a pretty nasty GI tract infection. Usually it's the result of taking a bunch of broad spectrum antibiotics. We call it Clostridium difficile. C. diff is what they just use. They shorten it to that. It's bad news. You've got some serious problems with your normal bacteria in your gut, and these bad guys, they basically take over because the good guys have been kind of depleted and wiped out. So now you've set the stage for some serious issues, serious illness as a result of that. And sometimes to treat it, to cure these people of this problem, they get a fecal transplant. They get feces from somebody and in turn transplant it into themselves, right? And they have found a couple of pretty interesting things in the process of doing this. They have taken an individual that needs the transplant, and this individual is overweight, and they get the feces from someone that is slim and trim, and that overweight individual, after they have been fixed of this problem, they drop a bunch of pounds. The bacteria that are within their gut change, and it ultimately changes them, right? And we've seen the opposite with somebody that was slim. They got a transplant from somebody that was overweight, and after the fact, they put on the pounds, right? It seems like, geez, could we have found this weight loss, this magic bullet, this magic nugget of poo that could make you slim and trim? I think most people probably would say, no, I don't want to do that, right? That's not the kind of sandwich I want to eat, right? I think you don't need to eat it, right? Kind of crazy, right? But it does fit right with this vial, right? What sorts of stuff do you have going on within your body, right? Okay, so to this, renewable fuels, there's all sorts of thought about what we could do. What sorts of things could we utilize that's not fossil fuels? And algae holds some promise, right? Are they a true solution? I don't know, but there's definitely some potentials with this, right? The algae, they're pretty good photosynthesizers, right? And in the process of photosynthesizing, you're pulling in carbon dioxide. And as you do this, using the energy from the sun, pulling in the CO2, you build a bunch of sugars, and you also pipe out some oxygen, right? Kind of a good deal, right? So if you take these algal cells and you give them excessive amounts of CO2, ample amounts of oxygen, but excessive CO2, it's just like, you know, you have found yourself overeating, right? And gaining some extra pounds. The algae does similar sort of an idea. With extra amounts of nutrients, it starts pumping out pretty good quantities of oils that it stores up as energy reserves for later on, right? These oils, lipids, are high energy molecules. And in turn, we can extract that and use this as biofuel, right? Biodiesel, or whatever you want to call it, right? They have made some jet fuel out of algae and the oils that they produce. So there's all sorts of thoughts as far as utilizing these things. Could we ramp this up? Could we harness enough energy? Maybe, right? Maybe not. It does take a tremendous amount of surface area in order for this to occur, right? I had a friend that he finished his master's here. He went down to Utah State for a PhD, and he was working with stuff kind of like this, some biofuels, and looking at initially here microbes for like bioremediation to clean up hazardous waste. But for the PhD, it shifted to more biofuel. And as he got to down to Utah to the lab, his advisor took him into this room that was an empty lab space. And he told him, you need to deal with this. You need to buy all of these, all this equipment, all of the stuff. You need to set it all up. This is going to be your deal, right? And he's telling me all of this, and he's like, it's like I just received this blank check, and I'm able to get whatever the heck I want to outfit this lab. I'm like, what? Awesome. This blank check was written by Exxon. Why? Because they want to know, right? What will we do, right? Once we have shifted from our thirst for fossil fuels and we transition, is there something that we could still be making money at That's really what they want to know, right? And they spent mass quantities of money to outfit this lab for algal research. That's what they're focusing on, at least they were a handful of years back. He has since moved on and is teaching at a university in Oregon right now. Not still doing this type of research, but it's pretty impressive, right? And there are all sorts of things from the military to corporations that are concerned about these details and what can we possibly do, right? So this image, not E. coli, this is a type of bacterium called Bacillus anthracis. This is the organism that causes anthrax. Here we can see the bacillus, the growing bacterium. There are a couple of them right here that are in the process of forming spores. These spores are the protective structures for that bacteria. Some bacteria can do it and some do not, right? So the bacillus makes these spores if the nutrients are running out or the temperature's too hot or too cold, something that makes it be like, oh, I'm gonna die. It starts to form these spores. All of the little kind of orange guys, the red things right here, these are all the spores, the protective structures for that bacteria, right? And the potential issues when we think about anthrax, right? Bacillus anthracis is a normal environmental bacterium. It depends on where you're at, but pretty likely you could go outside, poke around in the soil a little bit and probably be able to find yourself some anthrax, right? And grow it up if you were so inclined, all right? Oftentimes, some of the best spots for that would be at a farm, right? Especially something where you got livestock. So you go out into a corral where cattle have been, probably find some anthrax, right? It's just kind of there, right? There are all sorts of ways that you can grow bacteria up and truly get it. But once you've got it, seeing the spores helps to clue you in that we're looking at a specific type of microbe, all right? And then as we progress, this from CDC, as we start thinking about possibility of disease, these spores are often really the culprit, right? And there's all sorts of issues. We may get infected with it by coming in contact with something like soil or animal hides are a pretty good one. Sheep or cattle may have it on them. It's been called wool sorters disease because the sheep shearers sometimes get nicks, wounds on their hands as they're shearing the sheep. And the spores of the bacterium are also on the wool and it inoculates into the wound. It's a cutaneous form of an infection that's not typically life-threatening in those circumstances, right? It's the other stuff, if it gets into your circulation, into your blood, or you inhale these spores, weaponized grade anthrax, that's a completely different story, right? And pulmonary anthrax is a fatal, not always, but it can definitely be a fatal infection, not just this cutaneous thing, right? So with all sorts of ideas about manipulation and genetic modification, it sets the stage to think about other types of organisms and the potential to manipulate and modify some of those things, setting the stage for problems. So we mentioned plants as we showed the ears of corn and talked about just normal agricultural practices with that. But there are other thoughts as we start moving into laboratory applications to manipulate and modify things. These plums have been modified to resist plum pox virus. It makes me think of chicken pox, but I'm not sure exactly what the plum would look like with this pox virus. Probably not so appetizing, right? There are other instances with like tomato plants that have been manipulated with genes that allow them to tolerate much colder temperatures. So a little bit of a drop in temperature, a little nip of frost doesn't hurt those tomato plants. Depends on your circumstances, depends on what you may have grown, right? That sets the stage for some of these things and possible advantages. Also, maybe some disadvantages, right? There are things like canola that are used for cooking oils that have been modified with Roundup ready genes within them. Soybeans are a similar idea that allow these crops to be sprayed with Roundup, which is an herbicide, and it doesn't hurt those crops. It kills the weeds, right? And those seeds, they're controlled by these seed manufacturing companies that have manipulated and modified these crops. And if some of the seed winds up in a neighbor's field that doesn't have that same crop planted, they can get in trouble because they're growing this product that they didn't pay for. And it's all, it can even just be the bee that has pollinated the one plant and moved over and pollinated yours. And now you've got something transgenic in the mix. That's a problem. Definitely concerns with some of those things and who truly owns that. A little bit disturbing. So we showed at the start of this chapter the machine that Terry Mullis had developed, this thermocycling machine that allows a strand or a chunk of DNA to get replicated countless times, right? This is a different type of mechanism, right? This is something that's setting up for some proteomic patterns, and this is proteins, right? So we've got a slide, a pattern that's impregnated onto this, that's the specific messenger RNA that says, I want to build this protein. And we can take this and basically see what sorts of things are showing up. There are others that we call microarrays. It's got DNA sequences that are put on there. And it's a similar idea where we can see specific things that are turned on, turned off, that are used or not used, dependent upon specific stimuli, right? That may clue you in to details about something going on with that question. This for cancer, as we look at this. So I want to tell you a different story, not cancer, right? There are and were some researchers across the way, because a couple of them have left, all right? They were looking at a couple of things, right? But the initial thing that they were looking at was pharmaceutical drugs, psychoactive drugs, stuff like Prozac and other things like that, similar nature, that people take. And when you go to the bathroom, lots of these drugs are cleared in the urine. So you void them out in your pee, right? That stuff makes its way to the waste treatment plant. But the waste treatment plant doesn't remove that type of stuff. That's not what it's doing. It's getting rid of microbes. It's getting rid of harmful pathogens. But the drugs that are potentially diluted and dissolved within your fluids are there, right? And they're not removed. That water winds up in the river. It winds up in waterways, right? And the question is, what do these psychoactive chemicals do? What impacts do they have on the environment, right? What do they do to the fish is really the deep down question, right? And they have noticed that there are handfuls of these chemicals that are triggering a sex change or sex shift in these fish, right? Where they go from male to female or female to male, one way or the other. Sometimes fish can do this just normally, right? Which is kind of crazy in and of itself, right? But through chemical means, chemical manipulations, they have seen this stuff occurring. And that raises some pretty serious flags, right? What's happening? And then that begs the question, if it alters fish, what does it do to us, right? What does it do to people that are consuming this stuff and they don't need Prozac, or they don't need whatever else it was? Or what if you are taking Prozac and now you're drinking it in your water too? Now you're taking more drug than you thought you were, right? What do all of these things do? So they find some of these issues, right? And then they start wondering, well, what does it mean for people, right? So it's not this exact same thing, but it's a similar sort of an idea where they start looking at these samples of water, they start looking at the fish, and they start sequencing the fish to see what sort of alterations does it have on their DNA. They run it through a microarray to evaluate the stuff that's turned on and turned off, and then they take that data and compare it to human data to see similar genes, similar proteins, similar expressions that are happening. What's happening in the fish, we can extrapolate and think about some of these things in people, right? And what they found was a bunch of things lighting up on the human side of it that all said autism. It doesn't mean that the consumption of this stuff in the water is going to give you autism, but it's a potential environmental link. It's a potential trigger that may help to stimulate that in the first place. We're truly not 100% with that, but we do think, autism has all sorts of things on its side, if you will, specific genetic predisposition, a specific sequence of genes that sets the stage to be more likely. And then we start thinking about environmental triggers and possibly some of these chemicals setting the stage for enhancing the likelihood. So then they start looking at the United States and trying to see what's happening in different areas of the United States. In Idaho, at least here, we drink groundwater, so well water. It's not the same as drinking surface water with giant reservoirs, where we can see that in other areas, right? Then they start asking these questions. What are the levels of autism with well water versus surface water? And the surface water that has excessive amounts of these chemicals in there, the levels of autism in these areas is considerably greater than levels where they're drinking well water, right? Autism still happens. We still have autism drinking well water, right? But lower than if you're drinking the other type of water. Again, it's not the answer, right? But it's a potential cause, right? That really makes you stop and wonder, right? What is, it's more than just what type of food are you eating, right? You think a glass of water is harmless, right? And you would hope that that is true, but maybe that's not necessarily the case. All right, guys, a little bit early, but I think that's a fine stopping point. Any questions or concerns? You're all thirsty now, thirsty for a nice glass of water. Okay, have a great day. Have a good weekend.